between the different chelicerae appendages. So we can identify chelicerae and pedipalps and walking legs across the different chelicerates, regardless of how modified or adapted they had become. But this is not true for the opistosomal appendages, where the homology statements here are much more challenging to establish. Some of those are unique to certain groups, such as like the spinnerets of spiders, and therefore it's very hard to establish what those structures are homologous to. In other cases, such as the book lungs, we're able to establish some level of homology. For example, all between the book lungs has internalized gills, or these later being derived into tracheal respiratory organs by positional homology, but it's much more challenging in general to do these homologies. And one of such abdominal appendages is that of the pectines. So pectines are structures that are restricted, are only found in the scorpions, and they have no clear homology with any other structure in any other chelicerate. There are hypotheses of what is the developmental origin of these structures. So one of those is that they were hypothesized to be epipodal derivatives that would make them deep homologous to the insect wings and also the gills in crustaceans. However, there is some evidence from the developmental research that suggests that these organs are instead of telopodal origin. So we have seen in natural occurring hemotic transformed mutants that the pectines can be in occasions transformed into leg derivatives with claws. So that point more to a telopod type of origin instead of a epipode. But beyond that, we still know very little of how to homologize these structures. So one of the things I'm trying to do in this talk is present you our results of our investigations of the gene expression in these appendages and how they compare or are diverse with other appendages in the scorpions. So we are looking at the makeup, the genetic makeup of these appendages from a comparative genomics point of view. And the organisms that we are basing our observations on is the Arizona bark scorpion, the Centroresus sculpturatus. And the reason why we choose this species is because it has a genome available, and this is reasonably well annotated. And this solves a lot of the problem to identifying where those genes are and what genes are these. And at the same time, it's a species that is easy to rear in the lab, and they can be bred in captivity with relative ease. And that is precisely what we did. We gave a series of adults in captivity for a period of time, and gravid females were identified and dissected, sacrificed, so we can extract the developing scorpion embryos from them. And we were specifically targeting for looking at embryos at the stage where the appendages are starting to develop. That is the limbot stage. And just this gives you a brief overview of the overall procedure. Let's say, for example, this is how a scorpion embryo at the limbot stage look like. And we dissected the individual appendage primordias. In this case, let's say that we're talking about the pedipalps. So we took out the pedipalps of these embryos and separated them into RNA intrisol for RNA extraction. And because these tissues are very small, we have to pull together the pedipalp primordia of different embryos to get enough tissue to extract RNA. And this particular sample was then used for RNA sequencing using an Illumina platform. And an interesting point here is that the sequencing was done in parallel in two independent lanes, so that for every single biological replicate, we had two technical replicates that represent the sequencing from the very same extraction. And to account for the biological variation, we repeated this procedure for each individual appendages 3 times. So that means that 3 different pooled samples were independently created and extracted each of these into different lanes. And we repeated this process for 6 embryonic appendages represented here, including from the prosomal appendages, the labrum, the chelicerae, the pedipalp, and walking leg 1. And from the pistosomal, we dissected the pectin primordia and the book-long primordia. All right, I'm not going to go into too much detail about the whole bioinformatic procedure here. I'm happy to answer any questions on details. I'll just mention very briefly that the approach that we use for quantifying and mapping these reads is based on a splice-aware alignment method and leverages on the annotation of the genome. And in general, for the whole differential gene expression analysis, we use two different approaches, HR and DSIC2. Just one of the first things that we can do once we have obtained the quantification of these genes is to conduct a ordination analysis to observe how the quantities of the genes vary across the different tissues. And this is done for a variety of reasons. It gives us a brief glimpse of the whole dynamics of the expression in all the tissues, but also allows us to spot technical errors. And one of the things that we see here, for example, is that the technical replicates are always very close to each other. So that means that they produce similar numbers, and also that the quantities from the same appendage tend to have very similar quantities of genes, expression levels, as they are more closely together. Very interestingly, the first principal component, the one that accounts for most of the variation, already separates prosomal from abdominal appendages. And we see that the pectins and the book longs are found to share very similar expression levels overall, so with respect to the other prosomal appendages. But this does not tell us about how these genes are differentially expressed. The previous one, the PCA, is just a broad overview of all expression of all genes. But instead, we are interested in identifying which are the genes that are more expressed in the pectin or less expressed in the pectin. And this is done in a pairwise manner. So all the differential gene expression analysis were done in pairs of appendages, all the time comparing it to the expression level of the pectins. And this is represented here in these volcano plots, the result of differential gene expression, in which every single dot here represents the expression level of a particular gene and their significance. And this is exactly what the y-axis tells us apart. Above this blue line here are those the genes whose expression level is considered by the differential gene expression program to be statistically significant at a significance level of 0.05. And also this graph allows us to see the direction of the differential gene expression. Those genes that are at the right side of the graph here are genes that are overexpressed, in this case in the pedipalp, and those genes in the negative part of the graph are genes that are underexpressed in the pedipalp and with higher expression in the pectin. I briefly mentioned that I used two different gene expression assessment pipelines, and the reason for doing that is that although these two tools have been shown to agree in great degree in their estimation of the magnitude of the differential gene expression, here represented by the comparison of the log2-fold changes in the two different methods that follows a clearly linear pattern, they tend to disagree in other respects, particularly with the level of statistical significance. And this is exactly what is, what is represented here. So instead of focusing in one or another, I'm gonna use or consider only as those differentially expressed, those genes that were identified by both methods. So in this case, represented by these purple dots. And this figure summarizes the results of all these comparisons. This looks like a complicated figure, but in reality, what it is a Venn diagram that summarizes the intersections of differentially gene expressed genes using this criteria across the different set of appendages and their differing intersections. Just very briefly, I'm just going to make a note here or bring your attention to the fact that the book long is the pairwise comparison that resulted in the least amount of differential gene expressed genes. That is, that most of the genes that are found in the booglong and the pectin show very similar expression dynamics compared to their appendages. So that also supports the idea that the booglong and the pectins are developmentally more similar. And this gives us a universe of genes that make the pectin different from these other appendages. But we could focus more interestingly in those that are shared across all these different comparisons. So this gives us a set of 69 genes that were found differentially expressed across all other appendages. And besides looking at which genes are these, we can also take a look at how is this expression moving on. So this is shown in this heat map. And briefly speaking, it shows the degree of expression of these different 69 genes across the different appendages. And here we can see highlighted the part that represents the pectins. So those forming a cluster in this heat map. And we can easily identify those genes that are highly expressed compared to the other ones. I'm going to skip to these slides just to briefly mention that after this analysis, we have a set of candidate genes that are probably controlling the development or specification of the pectins as a different organ. This list includes some known transcription factors that orchestrate the morphogenesis of these structures, but also there is a plentiful of genes of unknown function that are subject to future investigations. And with that, I'll conclude. And finally, I will thank my financing sources and my lab for all their support. Thank you.
Thank you, Jesus. If you would like to ask Jesus,
it is a shame that I cannot hear if there is questions or the silence is exactly what I should be hearing now.
Hang on, I can type it to you. Chat for questions.
Thank you, everybody, for your encouraging words.
Got about, if we're sticking strictly to the 15 minutes, we've got about a minute and a half left.
I think given the circumstances, we should, if anybody has questions for him, they can get a hold of him pretty easily. Can we
Any
go ahead and
chance
move
for
on
CRISPR? Yeah, well, we are hoping. I don't know, for scorpions, those are more difficult to work functionally than other arachnids in general.